Thank you everyone for tuning in to today's webinar. We're extremely excited to be able to learn more about the incredible advances in brain-computer interface technologies. And who better to share than our speakers from BrainGate, who I'll introduce momentarily. The team at BrainGate is working hard to help restore the communication, mobility, and independence of people with neurologic diseases like ALS. So we're thrilled to be able to hear this upcoming presentation. I also want to just make a quick shout out and thank you to the rest of my team at Unite for working behind the scenes to make this event happen. By way of background, my name is Tanner Dagdalen. I'm the co-founder and chief executive officer of Unite Genomics. And through the technologies that we're developing at Unite, we aim to rapidly increase the research community's understanding of ALS, get more patients into trials faster, and cut years off of the discovery cycle time for researchers. We envision a world where all patients are empowered to improve their health and no patient's disease goes untreated. And as far as we're concerned, this world is 100% possible. And as a community, we can make it happen. So today we're excited to have Dr. Lee Hochberg and his colleague, Dr. Daniel Rubin, with us. Dr. Hochberg will be presenting the details of some of the research that he has underway with Dr. Rubin as well on brain-computer interfaces. That presentation will be about 30 to 45 minutes, after which the three of us will have a follow-up discussion with Q&A. So if you have a question at any time during the event, please use the Q&A feature to submit your questions, and we'll reserve time at the end of the session to answer them. So now, without further ado, let me introduce our speakers. Dr. Lee Hochberg is one of the principal investigators on the BrainGate team and the director of the Center for Neurotechnology and Neurorecovery, Neurocritical Care, and Acute Stroke Services at the Department of Neurology at Massachusetts General Hospital. He's also a senior lecturer in neurology at Harvard Medical School, a professor of engineering at Brown University, and director of the Center for Neurorestoration and Neurotechnology at the Department of Veterans Affairs. He has with him his colleague, Dr. Daniel Rubin, who is not going to be presenting today, but he'll be answering some of the questions via text as you answer them and or as you ask them, and will also be part of the discussion at the end. Dr. Rubin is a clinical co-investigator at BrainGate and an assistant professor in neurology at Mass General Hospital, as well as an instructor of neurology at Harvard Medical School. So with that, thank you both for joining us today. If you're ready, we can go ahead and start your presentation.
Sure thing. Thanks, Tanner. Thanks so much for the really kind introduction and for the invitation to join you all today. Thanks to Unite Genomics for this opportunity. It really is a privilege for me to be with all of you today and to tell you a little bit about our ongoing BrainGate research towards the restoration of communication and mobility for people with ALS and for people with other disorders of the nervous system that can result in weakness or paralysis. Just as a brief disclosure, there's no personal financial disclosures to report. I am quite grateful for both the public and the philanthropic institutions that continue to support the ongoing BrainGate research, many of which are listed there on the screen. If I don't say it out loud later, I am going to be telling you about an ongoing clinical trial. It's an investigational device that I'm going to be telling you about, and the other disclosures that are listed there. The more important disclosure, I'd argue by far, are the great group of people who I get to work with every day. In order to do the research I'll be describing, it really takes a team that's across disciplines, across institutions, across the country. And many of you may be familiar on slides like this. It's you can pretty much ignore the people whose faces you see on the slide because it's everybody who's on the right side of the screen are students, fellows, research engineers, and everybody across these institutions who've really been doing all the work that I'm going to be presenting to you. So I'd like to begin with a reminder that both Dan and I spent some of our time in the neurointensive care unit helping people with ALS, helping people who have acute injuries of the nervous system. And when I think about people who I'm meeting, either out in their homes or in our intensive care unit, there's some simple goals that we have in mind. They're goals, they're mission statements in some ways for our BrainGate research. And for somebody with ALS, I would really like to be able to say with complete confidence that you will never lose the ability to communicate. And I'm going to be more precise about what I mean by that in just a little while. That is not something that we can say yet today as a clinical community or a research community. But as I think you'll hopefully agree by the end of the presentation, this is something that as a field, we're all getting much closer to. Similarly, for somebody who is admitted to our intensive care unit, who yesterday had no problem either moving or speaking, but suddenly, due to a brainstem stroke or other reasons that may result in what's up there as LIS or locked-in syndrome, I also want to be able to tell them that they will be able to communicate easily again tomorrow, not next week or next month or next year, but tomorrow. And for people with spinal cord injury or other types of stroke, I want to be able to assure them that they'll be able to move easily in a dexterous manner with their hands, for example, again, not in a while, but tomorrow. We haven't gotten there yet, but I believe that through the types of brain-computer interfaces, one of which I'll tell you about in some detail today, we as a research and a clinical research community are well on our way to achieving those goals. So I will spend one introduction slide or overview slide. I suspect that many of the folks listening today, many of you have heard about brain-computer interfaces, sometimes called neural interface systems. There are lots of them that have been designed that are currently in any number of clinical research ventures. And regardless of the specific BCI that you may be hearing about, all BCIs have three main components, which is a neural sensor, a decoder, and an assistive technology. So from a sky view, if we think about the brain of somebody who may be having difficulty moving for any of the reasons that we've mentioned, what we'd like to be able to do is to know how is that person trying to move. For example, if it's the result of ALS, if somebody's having difficulty or is unable to move their hand, move their right hand, we want to know that they're trying to move their right hand. And in order to do that, a brain-computer interface asks a few questions, says, what signals should we record from the brain? What parts of the brain, if we were to listen to the brain activity there, would tell us how that person wants to move their hand? And knowing the signal or the areas that we're recording from, then we choose a particular sensor. We choose a device of some sort to record that brain activity. To know, very loosely speaking, what is it that person's trying to do with their hand? Once we have that information, once we know that person's intent, the intention to move their hand, for example, we then put that into a black box. I will go into exactly no detail about that black box today. The equation that's up there is the only math that I think will appear at any point during the presentation. But in our particular study, which is the BrainGate research, what we're listening to, the signal that we're recording, are action potentials. They are the fundamental unit of communication between one neuron and another, one brain cell and another. And if we can understand the activity of lots of neurons, if we can know what that activity means, then we may be able to take that brain activity and convert it back into an intended motion, either of the hand or of a cursor on a computer screen. And sitting between those two is math, is computers that's converting brain activity into something useful. So before any brain-computer interface is developed, we have to think a little bit about what is it that BCI is going to re-enable. And there's lots of possibilities. For someone with limited mobility of their hands or an inability to move their hands, if we could restore free, rapid typing on a computer screen or a tablet computer, easy access to the internet, any number of text-to-speech applications, all of those technologies would likely be helpful. Also true for somebody who not only has a motoric inability of their hands, but is having difficulty speaking, that is either dysarthria or anarthria. If we can restore communication by any of these types of interventions and control that is controlled by that person, those would likely be helpful. There's lots of other technologies that are listed on there. For some hope for this research is that we're working towards reanimating control of the limb, that is re-enabling control of the arm and hand through any number of systems as well. So what's on the screen, the only block diagram I think I'll show is what a BCI is. So if you hear about any of them, they all have these components. You're recording from the brain, you're figuring out what the brain is trying to do, which of course really means what that person is trying to do, and then hopefully allowing them to do it. So skipping ahead a good bit, the technology that I'm going to focus on mostly today, and all of the data, all the results that I'm going to show you are from this item here. And I believe you can see my cursor. That is a little array, or what I'll call array of electrodes. It looks like a tiny hairbrush. I'll actually show you one of these in just a moment. I don't know how well it's going to show on Zoom, but I'll try. It's 4 by 4 millimeters, so it's baby aspirin sized. And each of those little times that you see, if one of those arrays is tapped into the brain, there's 100 of those little times, 96 of them are active. One's able to then record, or has the potential to record, from dozens, perhaps 100, sometimes even more, individual neurons in a part of the brain that in some way is related to the voluntary control of movement. And I have cavalierly skipped over 40 years of publicly funded fundamental neuroscience research that showed that it was possible to record in neuroscience laboratories in the university from the brain, and to be able to begin to understand a little bit of what that brain activity means. And it was based on that decades of fundamental research that we started the BrainGate clinical trials in 2004, asking what, at least at first, is a simple question. Is it safe and is it feasible for a person with ALS, brainstem stroke, or spinal cord injury to have one of these devices in place and to control an external device such as a cursor on a computer screen simply by thinking about the movement of their own hand? As I'll show you again in a moment, this device is connected by some fine wires today to a pedestal that's protruding from the top of the head. And at least in this diagram, that pedestal can be connected to a cable, which goes to some other amplifiers and computers. And if everything works the way that we want it to, somebody who's unable to use their hand thinks about using their own hand, or intends to use their own hand, can control a cursor on a tablet computer or elsewhere. This was pretty much what the block diagram or the illustration looked like in 2004 when we started. Since then, there have been 14 participants in the BrainGate trial. The rest of these numbers here represent, particularly towards the top, other outstanding research groups around the country that are using very similar devices, or the same device, and doing very similar research. And this type of brain-computer interface research with an implanted neural sensor, or an implanted device that can record neural activity, is now going on around the world. So based on this ongoing BrainGate research, happily, there are lots of different signals that can be recorded from different parts of the brain, and lots of groups around the country and now around the world that are learning more and more about how to convert, as we informally say sometimes, thought into action, or the intention to move into control of an external device. From a clinical research standpoint, which is how we all often hear about clinical research studies to begin with, the participants that we are recruiting will have limited use or decreasing use of their arms and hands due to either spinal cord injury, stroke, muscular dystrophy, or focus for today on motor neuron disease, ALS, or others. People recruited to the study are adults who are at least a year out from their injury, or at least several months out from diagnosis. Participants in the study do need to be able to communicate, but they don't need to be able to speak. We have had participants in our study with locked-in syndrome who are communicating undoubtedly slowly, but fully with their eyes, asking questions and interacting. And so they're people with, for example, ALS, who are having difficulty speaking can very much, or in the inability to speak, can join us in the trial. Similarly, people can be mechanically ventilated in the trial as well, although people can join us earlier on as well. Otherwise healthy, this point about living within 3 hours of the study site, the reason for that is much of the research, nearly all of it, happens in the person's living room, wherever they live, maybe their living room, maybe in their bedroom if they're sitting up or laying down in bed, or it may be in an assisted care facility. While, of course, there's a neurosurgical procedure that is required in order to place the device, which of course happens in the hospital, in an operating room, After the participant returns home, the rest of the research, all of the data that I'll show you has been collected wherever that person lives. Which is what I just mentioned. In that point, we are going to the participant's home. And this is an ongoing research that's happening both in Boston, Providence, in Cleveland, and Palo Alto as well. Let me do two things with this slide in the background. I'm going to do what I'm sure is going to be difficult, especially because I'm not seeing myself on Zoom at the moment. I'm going to hold up one of these devices. I'm going to wave it around, hoping that you can see it a little bit. The goal is sort of to point out that it's small, and I'm going to try to bring it up close to the camera for just a moment. You might briefly see that there are a whole bunch of little times on this essentially little hairbrush looking device. That is the array that's tapped into the top of the brain. This is the pedestal, and this is what enables the recording from these really powerful, informative, and kind of information-rich signals or neurons that are in the brain. The reason this research has made the progress that it has is because of the great collaboration across the institutions that are represented there on your screen. The MGH is at the center as the coordinating center, incredibly close collaborations with our colleagues at Brown, at the Providence VA on the two sides of the equation there, and at Case Western and Stanford as well. We're nearly daily all interacting with each other, if not multiple times per day. So what I want to show you now, and I'm going to see if I could just pause this video for a moment. I'm going to jump right to show you the very first participant with ALS who is in our clinical trial. And so I'm now jumping back to 2006. We see his chest there. You'll see the computer screen in just a moment when I press play again. And you see our research technician there, Abe Kaplan, who's telling him for the first time, telling our participant for the first time what he should be trying to do, which is trying to move his hand. I will tell you that the incredible gentleman who was in this trial was in his 30s, had advanced ALS. He was mechanically ventilated. He was unable to use his hands, with an occasional flicker remaining of 2 fingers on his right hand. He also had limited eye movement, but with that eye movement, he was able to use either another technology or to type out, if you will, letters, showing us not only his incredible sense of humor, but his real deep understanding of the research and desire to help other people with ALS and other forms of paralysis. So as I press play, he's being asked for the very first time to try to move that little white cursor on the computer screen and to move it to the orange square. And I purposely don't have the sound going at the moment so that you can hear me, but you may have seen Abe, our research engineer, there smile, kind of looking around behind him, because even before the instruction to move that cursor was fully spoken, our incredible participant was thinking about moving his own hand. We were decoding the brain activity, and he was moving, albeit in a wobbly manner, but kind of right out of the box, if you will, that cursor around the screen to get what were the big targets. That told us something really important from the standpoint of ALS, because there was a question, maybe that's a question that may be asked, why would we record from the motor cortex from somebody who has advanced ALS? Because there are some people that were under the impression that because ALS can injure the motor cortex, that we wouldn't get any useful recordings. The good news is that our experience thus far has been that there's lots of useful, harnessable, if that's a word, activity in the motor cortex that we can allow people with ALS to use to control an external device. You're now having the perspective of one of the users of the BrainGate system in our trial. He's looking at the computer screen. This is somebody, again, with ALS, and he has neural control, that is intended motion control of that cursor on the screen, and he's moving it to the red dot. This is a video game, and when he gets there, he's supposed to click. So for the movement is an intended movement of his hand, as though he had his hand on a computer mouse, and when he got to the target of interest, it was a click. A few things to point out about this. One is that compared to the first video that I showed you, which was from our first participant, to the one I just showed you now, the control of the computer cursor has substantially improved since then, which is the result of the ongoing research to improve that black box, to improve the math, to improve the decoding ability of signals from the nervous system being used, from the brain being used to control an external device. So smoother, faster, more accurate. The other thing to point out is the background, which is that we're in his living room. Those are his paintings, his couch. So this research really does occur at home. Ask why are we doing this in people's homes as opposed to the university laboratory, which is where research usually happens. The answer is pretty simple, which is that a medical device needs to work not in the university laboratory, it needs to work where people live. And since the beginning of this research, we've been conducting it with the help of extraordinary participants who have allowed us into their homes to work with them for a year or more, and have shown us what it takes to have this type of brain-computer interface work where it really needs to, which is at home and eventually outside of the home as well. So when we saw just a few years ago this quality of point and click by somebody with limited or no use of their hands, we said, okay, we now need to get ever better at various types of control of other systems. And I'm going to come back in just a moment to communication systems. What I want to show you now, which is a video that I know that some people have seen, just stepping back a little bit. Even though communication is much of the focus of what we're doing, control over robotic systems that permit environmental control is also a portion of this research. The woman that you're looking at about 15 years prior to this video had a brainstem stroke. She did not regain the ability to speak or any functional use of her hands. But here we're looking at a video of her more than 5 years after joining the trial, still recording useful signals from the part of the brain where we had placed the array. She was thinking about using her own arm and hand to reach out and grab that thermos of what I'll tell you is a cinnamon latte for scientific precision, and to kind of reach out and grab that thermos and then bring it to her mouth. She here has a restricted sip power, so it will take her a little while to be able to get that cinnamon latte into her mouth. But you saw the beginnings of the smile as she was able to do that. The reason for that, and we'll see, I think in just a moment or two, when she's able to get that drink into her mouth, her reaction to being able to do so. And when we see that, that's not because it had been a while since she had had something to drink, as much as it had been a while, meaning more than a decade, since she was really able to do that solely of her own volition. That is, to move something in the environment, whether it's a thermos of coffee or anything else that she wanted to move from one location to another. Her ability to do that just by thinking about it really was it was an incredible moment for her and for us as well. Getting back to the point and click control over a computer cursor, when I press play here, there's going to be somebody who also somebody with ALS who's looking at the computer screen that we're looking at. We can focus on this one here. There's another example of somebody else with ALS in the upper right-hand corner using a different type of computer interface. Here, there's kind of a funny looking keyboard. It's actually an optimized keyboard where most commonly used letters in the American English alphabet, at least, are right down the center. And here, one of our participants with ALS is pointing and clicking to type the phrase that you see at the top of the computer screen. And as shown on the left, she and others in the trial were able to achieve up to 39 correct characters per minute just by typing, and that's without any word prediction. One of the reasons that we're working on a system that records from inside of the brain is that we believe, and so far it seems to be, that those information-rich signals can provide much faster methods of communication than other approaches. Of course, other approaches may not require or do not require neurosurgery, and that's an important balance. But when we saw this speed of typing, we began to think to ourselves, well, this is pretty good point and click control. It's pretty fast and it's pretty accurate. why are we, as a group of clinicians and scientists, why are we developing communication interfaces? Why don't we switch to using communication interfaces that are, if you will, professionally made? That is tablet computers. There's an entire industry that's focused on doing this. You're looking at two people with ALS. A woman on the left has one array in the brain, and therefore one of these protruding, small protruding pedestals and a not so small looking cable that's connected to the rest of the computers behind her. I'll show you how we get rid of that big cable in just a moment. The gentleman on the right has two arrays in the brain, and therefore two of the pedestals and two of the cables. She is typing an email that is an unmodified tablet computer using not assistive technology, but a communication technology, that Google Nexus tablet, if I remember correctly, that she's using to type to one of our researchers. The gentleman on the right, that's YouTube, and he just did a search for ALS. So back to the beginning, when I said one of our hopes and one of our goals is to be able to tell somebody with ALS that they will never lose the ability to communicate. They will always maintain the ability to communicate. One of the ways that I mean that is maintaining the ability to control the same tablet computer as was being used before the onset of weakness. And I think that we're making some good progress on the way there. What you're looking at now is some decoded handwriting. So what that means is the letters you recognize, the blue squiggles you recognize as somewhat script letters, these are what I would call kind of the neural trajectories. The blue, well, I'll call squiggles, the blue characters are the representation of what we have decoded from the brain activity. As one of our participants here, a gentleman with cervical spinal cord injury, attempts to write each of those letters with his hand. And what that has enabled is, I'll show you, I think, two videos here. You see the gentleman on the right, you see the still cable that's protruding from the, that's been connected to the little pedestal on top of his head. And again, we've asked him to type or to handwrite or to attempt to handwrite the phrase that's on the top of the screen. And you begin to get a sense here for the speed at which he's able to do that. Maybe more importantly, rather than copying the phrase, is answering a question, an open-ended question, if you will. So we asked, where was he born? And this is the real-time appearance of the typed or handwritten or attempted handwritten phrases that as he's doing this. And here, achieving, the number of characters per minute that are listed there offline using language models can get up to 99% accuracy. This is work that was presented at as part, or at least preliminarily, as part of a brain-computer interface award symposium back earlier last year. And I think when we see this, and I get back to, we want to be able to assure people that they'll be able to communicate, it's by this type of rapid communication, whether it's point and click, control over a tablet computer, attempted handwriting. This is really one of the ways that we're focused on maintaining, restoring communication. Just a handful of slides left. I've shown you a few people in the ongoing BrainGate research with big cables that are connected to a small pedestal that's on top of the head. Those big cables are not a final form medical technology. Much like cardiac pacemakers or deep brain stimulators, at first there's a percutaneous device that allows the updates to be happening rather than in the body, outside the body, that is in the living room, and things to be updated and constantly improved. Happy to report that that cable, big cable, is no longer required. It can be replaced by something much smaller that can be placed on top of the head, connected to that pedestal. This is a wireless transmitter that we've been using recently, and that no longer needs to be placed by us on the research team. It can be and has been placed by caregivers allowing our participants who are interested in doing so to use the system at times in their own home, even when we're not really engaged in a research session, but instead allowing them to use the system, and importantly, to give us feedback. Is the system working the way that it's supposed to? How would they like us to make it better? What other technologies should we be trying to connect it to? And this is the type of key feedback that we get over the course of the study. And just an example of this being used, you see, realize it's a little blurry. This gentleman in the lower part of the screen laying on his right side. This is a fluorescent orange neck pillow that you're looking at. And really blurry here is one of these devices that is one of these small wireless transmitters attached to the pedestal on the top of his head. And as I press play here, we're looking at him. He's unable to move his hand. And that's just a Windows Surface tablet. And that is just Pandora, which in many ways is the point. It's going to go over and click the thumb.
Give it the thumb.
He's thinking about using his hand. He's thinking about moving and clicking and using that tablet computer much the same way he might have run his own hand over that tablet and tapped on the icons prior to his injury. So restoring communication, restoring environmental control. It's about us providing ever better neural control over the incredibly powerful communication and environmental control technologies that are available today, such as this tablet computer or others. And I could watch this video for hours, in part because it's simple. It's simple. He just switched to desktop to select. I think this was back in March, a few Marches ago, and watching the basketball game. So this is part of where we're headed as a brain gate research team, and really where the field of brain-computer interfaces is headed. Final step, not the one that we're at today, but already working in the university research lab, is a fully implanted system. That's where medical devices need to go. But in order to get there, we need the ongoing input from people who are enrolled in the trial, telling us what works, telling us what we need to continue to work on. And undoubtedly, these will be fully implanted devices in the hopefully not too distant future. So I want to make sure that I leave plenty of time for questions. This is an ongoing trial in both on East Coast and West Coast for people with ALS. Please feel free to, I know momentarily, to be asking questions live here today. You can email us at clinicaltrials@braingate.org. Again, most important slide, great group of people that I get to work with every day across a number of institutions. It's quite clear that this is a pre-2020 picture here. This is from our annual summit meeting in 2019, but it's really the people whose faces you see here that have been doing all the work and allowing us to make progress in this brain-gate, brain-computer interface research. So with that, I thank you so much for your attention, for tuning in today. I'm going to unshare the slides, and Tanner and Dan, I'll let you kind of guide from there. But thanks very much.
Thank you, Lee. That was amazing. I know that folks are going to be asking some questions in the Q&A box as we go here. So if you have any questions that you haven't asked yet, please submit those. But I have a few questions just to kick us off. So
sure.
One question is, and I'm sure it's on the mind of many of the people tuning in here, is, what do you think is the time frame by which this will be broadly available? Right now it's in research, and you participate in the research study to get access to it, but when is it going to be possible to just get the surgery or purchase something like this outside of a research context?
Yeah, and kind of jumping to the end, it's towards that goal that all of this research happens. And it's only with the ongoing research that ever happens, which I think everybody recognizes, but is really an important thing to, I guess, emphasize. A few years ago, in responding to that same question, I would have been measuring in a decade or more. I think at the point that we're at now, we're talking about years rather than decades. I think we're getting much closer to being able to say in a handful of years, maybe a few years, depending on one's definition of a few, it may be possible that one of these implanted brain-computer interfaces could complete its clinical trial safety testing, most importantly, and some demonstration of what, as I showed you today, I hope is the kind of basis for a valuable, useful technology that would help people with ALS or other forms of paralysis. So I really hope it's just a few years before we get there. And the first device that's out there, and I can't remember, you just used the word purchased or marketed, one of those, Tanner. That first device, I think, is going to do a lot. And then the next version is going to do even more. And it's going to be like any other medical technology that continues to improve over time. But one of the most incredibly rewarding parts of being involved in this research has been working with our incredible participants and having them tell us what's working and what's not. I can see a cursor moving on a computer screen quickly and accurately, and I can think that it looks great. But then I need to ask, so how was that? And did that work? Was that easy? Was that hard? And when people tell us not that it was easy, but that they just did it, and they can't even describe how they did it, then we know we're on the right track. And thankfully, people tell us that regularly. There are other times, though, that we, quote, improve our math, unquote, that is, improve the decoder, which is the research process. And it turned out we were wrong. We didn't actually make it better. We made it a little worse. And so then we go back to the drawing board, and the drawing board is back in the lab. And the following week, we're back with an improved, what we hope is, decoder. And it's that feedback really getting the impression of not only how people want to use the system, but what's working well, what we need to continue to work on that's been driving this research forward, not only in our BrainGate group, but I think kind of across the field.
And I want to dig into the mechanics of the trial, and I know there are lots of questions about the trial in particular, but just a quick follow-up on that. So are there, I think we've seen in the news companies like Neuralink, I think Elon Musk has been bringing a lot of attention to that. Are there any commercially available solutions that are trying to do something similar? Any companies that are putting, commercial budgets towards this sort of research too, or is it mostly academic research?
So you may have seen in the initial disclosure side that Mass General has a clinical research support agreement with 3 companies that are relevant in this space, if not more, which includes Neuralink, as you just mentioned, Paradromics, and Synchron. So I'm very fortunate to have the opportunity to interact with them every so often through the hospital, because what we're learning in our BrainGate research, I want them to know, because just as you said, with their budgets, with their corporate ability to be defining a system and moving it forward, I want them to learn and to know and to not have to learn everything that we've learned over the course of the trial. This is what publicly funded research is supposed to be, from the Department of Veterans Affairs, from the NIH, from philanthropy. We should learn as much as we can, and if we can hand that off to allow industry to define the product that will appear, that's what we want. And so I do think I'm excited that there are those companies and others that are involved in this research, and whether it's us or some combination of us and them in the future that allow these technologies to get out there. What we want is for people who are having difficulty with speech or movement or both, to have restored communication and mobility.
Definitely. And right now, that means participating in a trial, right?
Yes. And I'm looking at my phone, not to be rude, but to turn the lights back on. So there, that's better. Yeah. So yes, and to participate in the trial. I think
maybe we can dig a little deeper into the specifics of the trial. So I think you had a slide that kind of overviewed some of the inclusion and exclusion criteria for participation. I did have a quick follow-up question there, and I'm looking at some of the questions coming in too. Are there any other criteria besides those enrollment criteria that could, or any other factors that might preclude somebody from being able to participate in the study?
Yeah, thanks for asking. So like any clinical research study, there's a long list of inclusion and exclusion criteria. The general version is, particularly if we're focused for the moment up for people who have ALS, is the people who have ALS who are losing or have some weakness of the arms or hands. There doesn't need to be complete inability to move. People can be recruited to the trial earlier on. They can also have had the ALS for a longer period of time to the point that there's no movement of the arm or hands. In general, people need to be otherwise fairly healthy. They can have they can be using a mechanical ventilator, that's fine. But knowing that neurosurgery is a part of this, most importantly, we would want somebody to be healthy enough to be able to handle without real concern a few hour neurosurgical procedure. So really, this is just about weighing the safety of that elective procedure. And a lot of the focus is there. There may be somebody whose vision is severely impaired. We wouldn't be able to recruit to the trial because interacting with the computer in that case would be more difficult. So it's really on optimizing the participant's ability to use the system as maximally as possible for both the benefit of society, that is to allow everybody to learn as much as possible from that participant's engagement in the research, and also for the participant to be able to, we hope, use the system even when we're not there.
Definitely, and I think we're seeing lots of questions here about recruitment and enrollment. So are you recruiting patients now, and where can patients go to see what sites are recruiting, and if to check and see if they're close enough and that sort of thing?
Yeah, you bet. For people with ALS, currently recruiting is happening both in the Boston Providence area and in the Stanford Palo Alto area. We can draw kind of a 3-hour drive time circle around those cities, and that is our catchment area, if you will. So if you think you're right on the border of a 3-hour drive time, we will likely be able to take a look at that and say, yes, it's 3 hours. If it's a 4-hour drive time, probably not. And the reason for that is we're going to be in your homes, and we want to be able to get back and forth quickly enough and regularly enough that we can not only do the research with you in your home 2+ days per week, but also to have easy interaction with many of the other people that are involved in the study. So the best way to get in touch with us is through just sending us an email at clinicaltrials@braingate.org. That will get Dan to that website. If you happen to live in the Northern California area, we can then immediately forward that to a close colleague, Dr. Jamie Henderson, neurosurgeon and director of the clinical trial site for BrainGate out at Stanford. People may also be listening today either interested for themselves or for friends or family members with cervical spinal cord injury or stroke, inclusive of East Coast and West Coast. Our colleagues in Cleveland at the Case Western Reserve University are also actively recruiting for this and related studies.
And for a participant who doesn't end up getting enrolled, what is the experience like for them? So how frequently are they being visited? And let's start with that question. I have many follow-ups, and I think we're getting some coming in too.
Sure. Yeah, you bet. So the agreement, if you will, kind of going into the trial for somebody who joins us is, of course, the neurosurgery is happening in the hospital. Participant then returns home, and we're loosely agreeing that we'll have research sessions in the home, and I'll rephrase in the home in just a moment, 2 days a week for about a year, with each session being a few hours. So we generally reserve either morning or the afternoon or the middle of the day, based completely on the participant's own schedule. For us, certainly at first, to be physically present in the home, setting up the BrainGate system, working closely with that participant and with the computers that are nearby to make sure everything's working and providing immediate physical feedback to getting the system up and running. Because we do have the ability to use the wireless transmitter that I showed you, as well as to allow the participant to use the system when we're not there, we can conduct much of this research now remotely. That is, with us dialing in to access those same computers that the participant is using, that is the same BrainGate computers that the participant's using, and interacting and doing some of this research over Zoom. But at our first pass, we want everybody to be comfortable with the possibility of us appearing and kind of scheduled appearances, of course, in the home about 2 days a week for about 3-ish hours or so from kind of walking in the door to leaving for about a year. At the end of that year, I should mention, the reason that we say a year is that it's good to have an initial pausing point. And at the end of the year, we say, okay, that was a great year. We learned a lot. Do you want to continue? And so long as person wants to continue and we have all of the kind of regulatory permission and financial support for continuing the research, then we continue, and nearly all of our participants have elected, that could to continue beyond a year. And I showed you the video of one of the women in the trial who was in for more than 5 years. And we actually expect that people will be in the trial for 2, 3, 4+ years now.
Wow. And you mentioned that they're able to use the system even when you're not there. Does that mean that they can use it for non-research purposes, in between visits and that sort of thing? So it's available to them all the time, or?
So the goal is for it to be available for them all the time. Actually making that work is part of the research. So, the first part of the question is absolutely, we want it to work when we're not there. And ideally, we want to be told tomorrow that you used it today and to be pleasantly surprised by that. We're almost there right now, where people are certainly using it at home unrelated to any actual direct research session. We're not learning more about the brain necessarily at that moment. We're not learning more about the neural signals. We're allowing people to use the system to control their tablet computer and learning how well it works, and then finding out what the glitches are along the way and trying to make them better for the following week, which is why there's a big lab of any number of groups of the people who you saw in that slide earlier, who are updating neural decoders and constantly updating the software, so there can be easy, common neural control over an external device. One of the many kind of reminders that I kind of have for people who are considering the trial is the reminder that this is research, and that if it worked all of the time, it would no longer be research. It would be the product that Tanner, you mentioned earlier. But it's only with the incredible feedback that we get from people who join us in the trial that we're able to develop a system that's going to help other people with ALS in the future.
And what is the maximum capacity of the trial in terms of enrollment? So is there a limit, like a per site limit to how many patients can be enrolled at the same time, or?
There's not a per site limit. The current BrainGate trial has permission to recruit up to 21 people in the trial. At any given site, we have had as many, a site being a city where one of the research labs is, we've had, I think, as many as 3, which doesn't sound like a lot, I realize, people simultaneously. More commonly that we have either 1 or 2. And the reason that those numbers seem small is there's 2 reasons. One is just how much we learn from each person who joins us in the trial. It's what has enabled, with only 14 people having been in the trial over the past 15 or so years, the reason we've gotten as far as we have is that each person works with us so closely and is able to teach us so much about advancing the system that that interaction, which in addition to being incredibly rewarding for me in terms of the just learning from people who have had ALS, spinal cord injury, stroke, and how they would use the system, but also just in advancing the science of neural decoding and understanding how this part of the brain works to help people with paralysis. So that's one reason. The other reason is the flip side of that is we want to be available 2+ days a week in the home to each person that's enrolled in the trial. And once we get to about 3 people locally, our ability to keep up the way that we want to and have the full dedication to each of the participants who so wonderfully joined us, it begins to, we can get a little bit stretched thin. I look forward to expanding even further, but right now, for people with ALS on both coasts, we are recruiting.
Excellent. So do you have any idea of how many people you're looking to recruit right here and now? Let's say, if 100 people came your way today, how many of them would you try to get into the trial?
So I try for 100. I'm not allowed to do that. We would start with 1. And then very soon after that, measured in months, I hope it would be the next person. There's also, I should point out that clearly there's a neurosurgery that's involved. There are risks that are involved in neurosurgery. I wanted to make sure that we describe what was possible from a research standpoint in this trial. Clearly there are risks that are important to have a deep discussion of, and not, whether it's here on the web or in person, not a one-time discussion, but multiple discussions and a real kind of interaction over a period of many weeks to make sure that we're answering as many questions as possible, that people are thinking of as many questions that are pertinent to them as possible. And even at that point, then we're agreeing, that is consenting to the trial, and we're doing additional research, having imaging, CAT scan or MRI or both of the brain, and then scheduling surgery. So this, the idea of having 100 people in the trial, I can't wait. I think that's the right way to be doing this. We're going to continue to proceed single digits and working with just great people and learning together as much as we can, and having a handful that are active in the trial at any moment.
And so you mentioned some of the risks that you'll be talking through with any potential participant or anybody who's interested. Can you go over just kind of at a broad level some of the risks that people should be considering? I think there's a couple questions here. One is, long-term consequences of the implants, whether it's neuroinflammation or glial scarring. We have a question, a very specific technical question there, or risks involved with the surgery and during the trial.
Sure. Why don't we start with the clinical question? I am very blunt about and plain about the surgical risks. And I tell everybody that every surgery, including this one, has a small but non-zero risk of death. It's surgery. It involves general anesthesia, and I can't make that risk zero, and nobody else can either. If we thought that was going to happen, we wouldn't be doing this research. But when it starts with that possibility that there is a very small but important risk to consider, and this is not a finished product, there's no benefit that I am claiming is the result of being involved in this research. The benefit is not to the individual. The benefit is to helping us to develop and test a technology that will help other people with paralysis, other people with ALS. And it's kind of that equation. The people who join us are people who really want to help other people with paralysis and other people with ALS in the future. And so those are the incredible people who join us and accept those risks and move forward. Of course, Dan, the rest of the team, our neurosurgeons, all of our job is to minimize those risks as best as we can and to explain them. Having mentioned that one, the other ones that we really think about are the risk of stroke, seizure, bleeding, and infection. Some of those, if they happened and they were small, would be of very little to no consequence. With anybody who's in the trial, this is a much more in-depth conversation. So I'm going to, not because they're not important, but I'm going to give the quick overview now. The one that is actually on my mind all of the time is the risk of infection. Because there's an array in the brain, there's a device that's protruding through the skin, and there's some wires that connect those two. So we, from the outset of this, have been as careful as we know how to be in planning the surgery and minimizing the risk of infection. And there's also a skincare protocol that the caregiver, whether it be mom, dad, brother, sister, spouse, children, that help us to keep this part of the skin where the pedestal or pedestals are protruding through to keep it clean. So we use an antibacterial soap. There's also an antibacterial cloth that looks like a little cloth donut that's impregnated with an antibacterial solution, chlorhexidine, that's placed around that pedestal. And again, if we thought the risk of infection was high, we wouldn't be doing this. And knock on the wooden table that I have here, we have not had any infections in the nervous system, which is, of course, what we're eagerly trying to prevent thus far in the trial. I hope that it doesn't happen. I don't expect that it would happen, but it is a risk, and that's why we spend so much time making sure that we have a protocol to minimize that risk.
And longer term, I mean, obviously the research has been ongoing for 14, 15 years. What was the longest a patient has been a participant?
5.4 years, 1,994 days.
Okay.
I mentioned that for two reasons. One is that the woman who was in the trial for 5.4 years. The reason she decided to no longer be in the trial was that she was moving, and she and the equipment were going to be living in a different place. And after 5.4 years, she said, "This was great. I think I'm done." And we said, "Thank you." Over and over again, just for how much she taught, not just us, but that she really taught the world about the potential for these technologies. And so we looked at the calendar, and so we happened to know that was just shy of 2,000 days, or over 5 years.
And I'll just ask one other question, because I know we're coming up against the hour here. So, and it's a follow-up to the long-term risks.
Yeah.
Are there any other consequences of having the chip installed long term? So after the trial, does it remain installed? Are there any consequences, like not being able to have MRIs, or able to go through certain is it going to get picked up by metal detectors and that sort of thing that people should be aware of?
All good questions. So our recommendation is that at the end of the trial, that the device be removed. As long as the full device is removed, there's no preclusion to having an MRI. While you're in the trial, we tell people that they cannot have an MRI. So that's the approach. And I think would be the approach for the future. Now, at the end of the trial, when somebody says, "This has been great," hopefully they say, "This has been great and I'm done," the decision of whether the device should be removed is not up to us. That's up, we kind of informally will say, once you have a device implanted in the brain, it's yours to keep, and nobody's going to remove it without you saying that that's okay. Which just to state the obvious, perhaps. But our recommendation is, if the device is no longer being used, that we return to the OR and have it removed. And as long as all of the array and the wires that are there are all fully removed, then the person would be able to have an MRI again.
Got it. Okay, well, we have some additional questions here that we don't have time for. I just want to let all of the attendees know that we are recording all the questions, and to the extent that we can answer those questions offline and send a kind of brief Q&A or frequently asked questions document out with the recording of this, we will. So we will get to your questions, even if we weren't able to ask them here. This concludes today's discussion, and I just want to say thank you again for joining us. A very special thank you to Dr. Hochberg and Dr. Rubin for being here and answering all our questions. All the attendees will be receiving a post-event evaluation form in your emails, and we'd appreciate it if you could complete it to help us improve these events in the future. We also appreciate everyone's support in general, and look forward to all of us working together to advance ALS research and treatments. One of the things that Dr. Hochberg and also Dr. Rubin mentioned in answering some of the questions is that there is plenty of opportunities to participate right now. There's lots of opportunities that will open up in the future as well. And I know that both of you mentioned that there's an email that people can send an email to just get in contact and open up that dialogue. We'll make sure to send out the information so that people have that email address and they can email you as well. Is there anything else that you wanted to mention to the attendees about how they could participate, or anything along those lines?
I want to thank again for the opportunity to join you all today. The website's braingate.org. The email addresses are there. More information about the trial is there, and clinical trials at braingate.org. Happy to answer any additional questions people who may be interested. We'd love to hear from you. And most importantly, the next step is just continuing the discussion. There's a lot more information to share. There's more about the trial. There's more demonstrations of people using the devices in various ways. And even if you're just thinking about it, we'd be very happy to share additional information with you. And I thank everybody for joining us today.
And as always, the time to act is now. So we will send out the emails, the email address that you can use and get in touch as soon as possible.
Great.
All right. Thank you, everyone.
Thank you. Take care.
Thanks.